Okay, shall we continue, guys? All right, so far we have seen how reverse transcription happens in these retroviruses. But one thing to add on is that the reverse transcriptase actually lacks the 3' to 5' exonuclease activity. So what that means is it cannot really proofread the newly synthesized strands. Cellular polymerases has this activity, the 3' to 5' exonuclease activity, but the reverse transcriptase lacks that activity. And as a result, there's a relatively high mutation rate in these viruses. So there's about anywhere from 1 to 10 incorrect nucleotides per proviral DNA molecule. Okay? So as a result, this retroviral population is not considered a uniform population. So far, we have seen viruses that show us a uniform population. But in this case, because of these mutations, we do not get uniform population. It's called quasi-species, which is basically a collection of variants due to this accumulation of small mutations. Okay? Now, the linear double-stranded DNA, it remains associated with the components of the viral core. So what that means is all of this is still happening inside the viral core. You haven't even gotten inside the nucleus yet. Okay? So the reverse transcription reaction, etc., it all happens in the viral core. Okay? Then you form what's called the pre-integration complex. This is a complex of your retrovirus capsid proteins and newly synthesized proviral DNA. And this is now ready to be integrated into the host genome. But the problem with this is too big to go through the nuclear pore. So most retroviruses actually wait until the cell divides and the nuclear envelope disintegrates. Okay? So in this case, the genome will only get integrated in dividing cells. HIV uses a different mechanism to get in, but other retroviruses, most of them, they wait until the cell divides, the nuclear envelope disintegrates, and then it integrates its genome into the host genome. And as you can expect, this integration is done by integrase that's also packaged in the core of the virion. So inside the pre-integration complex, you also have the integrase. Now the integrase, it will bind to the two ends of the proviral DNA. It will tend to bring them close together. And now this proviral DNA gets integrated into the cell DNA at random sites. It's not like it goes to any one specific site. This genome can integrate at random sites. Now, the integrase actually has to remove 2 3' nucleotides on each viral DNA strand that leaves a free 3' OH. Why? Why is this important? Why do we need to have a 3' OH on these strands? Because you need to form the phosphodiester bond. Okay? So that OH will bind to the phosphate to actually integrate into the host genome. Okay? So the way it works is that the integrase enzyme, it basically facilitates a cleavage and a ligation reaction to join your viral and cellular DNA together by bringing these OHs close to the host DNA to make your phosphodiester linkages. Okay? So this is usually about 4 to 6 nucleotides apart on the host DNA. So as you can see here, here's the host DNA. So what needs to happen is that the integrase needs to cleave the host DNA, because remember, the strands are joined. You need to break the strands to actually insert foreign DNA, or the viral DNA in this case, into the host genome. And that's done through your 3' OH forming the phosphodiester linkages. Okay? But the problem with this is this cutting and joining, it leaves a single-stranded gap on the host DNA, which is about 4 to 6 nucleotides. And you also have 2 nucleotides unpaired gap on the viral DNA. So you can see that here. Here's a little bit of gap here, and here's a little bit of gap here. Okay? But our host DNA does not like these kind of gaps, so the host repair enzymes kick in because they think it's DNA damage, so they carry out repair synthesis of these gaps and remove the unpaired nucleotides of the viral DNA. And as a result, it also generates a direct repeat in the host DNA. Okay? So it's basically a repeated sequence that's present on either end. So have a look at this sequence here and this sequence here. Because you're joining it together and you're filling in the gaps, you're going to fill the gaps with complementary nucleotides. And as a result, it's going to generate these repeats in the host DNA. So once this genome gets integrated, your proviral DNA becomes part of the host genome, and as the host genome gets replicated, it gets replicated. So as the cell replicates the DNA and divides, all the daughter cells will now have this integration of the viral DNA. Now, the spread of infection can happen in different ways. So you can be infected with progeny virus, could be dividing cells that carry the proviral DNA. They will give that to the daughter cells as well. And they can be transmitted from parent to offspring because of DNA integration. Okay? So if that DNA integration is in the germ cells, obviously it's going to get transferred to the progeny. Okay? So for example, you have somatic infection, you only get horizontal transmission. But if there's a germline infection, you also get vertical transmission. So what that means is their children will have the proviral DNA and they will eventually develop AIDS. Okay? Now, we have gotten fairly good with actually treating HIV symptoms, and we have gotten to a point where some of these reverse transcriptase inhibitors, etc., yes, you have to take that medication throughout your life, but you get good quality of life. Okay, you don't tend to develop disease symptoms. But a cure itself will require the removal of all the integrated viral DNA from the host. This is not currently possible. There are some clinical trials and some in vitro studies that have edited out the HIV genome, but imagine editing that out in every single cell. That could be very, very problematic. Yeah?
Would it use something like CRISPR to take them out?
So yes, in the lab you can use CRISPR, and there have been some trials in patients as well. Problem with CRISPR is it sometimes has off-target deletions, and that can actually really cause major issues. So it's not fully there yet, but the retroviral therapy that people are using to prevent spread of infection and also to prevent development of full-blown disease, those work really well. Okay? All right, so far we have seen entry into the cell, how the reverse transcription happens, and how integration happens. Now, next thing we're going to look at is transcription and splicing. So this is the late phase of your replication where your viral RNA and protein synthesis takes place, and you assemble the virions as well. Your proviral DNA becomes unexpressed, or it remains unexpressed or latent for fairly long periods of time, as we saw earlier, because it needs appropriate transcription factors to actually make those proteins. Okay? Now, the U3 region that we saw earlier, it has specific sequences that actually interact with the cellular machinery. and that controls how the transcription takes place. Under what conditions does the transcription take place? Okay? And in addition to all that, you have your sequence elements in the terminal repeats that direct the transcription, as well as polyadenylation by the host cell enzymes. So again, let's have a look at the figure. Here's your proviral DNA. You can have your transcription, capping, cleavage, as well as polyadenylation, all done by the host cells. Okay? So you make full-length RNA, and you can also make spliced RNA to make your envelope proteins. Okay? Now, another thing to add on to this is that for transcription, what we need is a Tata box. Okay, so remember that's a consensus sequence that's present where? Sorry? In eukaryotes, yes, but where exactly? Okay, at the beginning of the transcription, what is that region called? The promoter region. Okay, good. So it's present in or close to the promoter region. You have your U3 and R junction, so the repeated sequence junction on the left, and this directs the transcription by your RNA polymerase, your cellular RNA polymerase. And we can use cellular RNA polymerase because the genome is now integrated and it's a DNA molecule. You have a poly-A signal on the right long-term or long terminal repeats, so between your R and U5, which directs the cleavage of the transcript at that particular boundary, and your poly-A then gets added by the host cell enzymes. Okay, so there's a cleavage sequence here, and then you add the poly-A tail when you make the RNA. This generates basically full-length RNA molecule that's identical to the genomic RNA of the virus. Okay? And all retroviruses, they make at least 2 mRNAs, but they can be more complex. The 2 mRNAs is very simplified version. You have your unspliced full-length RNA, which makes GAG and GAG-POL proteins, and the spliced version encodes for envelope proteins. Okay? So you have your GAG proteins and your polymerase proteins from the full-length RNA, and from the splice site you make your, once it's spliced, you make your envelope proteins as well. All right, so far we have seen reverse transcription, integration, transcription, and splicing. What happens next? Well, next comes assembly and release. Your virions will mature into infectious particles after they bud off from the plasma membrane. Okay? But before that happens, your envelope proteins get translocated into the lumen of ER, and then they go through Golgi and direct to plasma membrane. Why is that? Why does it have to go through ER and Golgi? What happens? What happens in ER and Golgi? Anyone? Yeah. Okay, you have to fold the protein to the correct conformation, yes, but you also have to modify the protein like glycosylation. Okay? So there's lots of modifications going on these proteins as well. Your GAG and POL polyproteins that are present in the cytosol, they will initiate the assembly of the viral core. So here's your RNA polymerase II carrying out transcription as well as splicing. You send the RNA out of the nucleus where it gets translated into your various proteins, and it will start assembling at the plasma membrane. And here on this end, you have your envelope protein going through the Golgi, etc., and from there, it will merge into your budding particle. Okay? Now, couple of things to add on here. Your GAG protein is targeted to plasma membrane by myristate, which is a lipid modification, and that allows the protein to insert itself into the membrane. Your nucleocapsid portion binds to the packaging signal. So remember that size signal that we talked about earlier, which is right here. So this is where the nucleocapsid binds to the packaging signal on the genomic RNA, and the protease will get activated and cleave your polyprotein into a mature single set of proteins. This happens after assembly. So here's your GAG assembly, your viral RNA, and finally your virus will bud off, and it will mature and then bud off from the cell. Okay? Everyone clear on all this? Yes or no? I know it's a lot of information, so please make sure you guys go and review this. All right, so now we have seen what a life cycle of a retrovirus looks like. Now let's specifically look at HIV. HIV is, again, single-stranded RNA molecule. It's a linear genome, and it's positive sense. There are 2 identical copies of RNA in each virion, as well as you have the tRNA that's specific for lysine. Okay, this is all packaged in the virion to be used as primers for reverse transcription. But what we see is HIV is actually a fairly complex virus. Okay, so you have your various HIV structural proteins that we have already seen, but you also have your various non-structural protein. Okay, we haven't really seen the virion protein R, but it is present in HIV, and then you have your other non-structural proteins. Okay, and you can see where these proteins are in this schematic. So what does the genome look like, and what proteins do you encode? You have 4 capsid proteins, so matrix, CA, matrix capsid, nucleocapsid, and a P6 protein, also called the budding protein or BP. Okay, so the GAG part here in HIV is these 3 proteins. You have your 3 enzymes: reverse transcriptase, integrase, and your polymerase. Okay, not polymerase, sorry, your PR enzyme, protease. And you have your 2 envelope proteins, your GP120 and GP41. Okay, surface protein and a transmembrane protein. And after that, you have 6 different regulatory proteins that you can see here. So here's your long terminal repeats, your various U3R and U5 sequences. Here's the size sequence, GAG, polymerase, and your various non-structural proteins, plus your envelope, and again, some non-structural proteins. And then you also have the 3' long terminal repeats. Okay? And you can see the same proteins in this schematic. Okay? So as you can see, there are multiple proteins involved, so it's a fairly complex virus. And it also has a very complex splicing pattern for these additional proteins. Okay? Because remember, HIV-1 is making all these additional proteins that we didn't see in other retroviruses. So because it has all these extra proteins, it makes those proteins through these complex splicing patterns. So you have your structural proteins, your GAG, polymerase, envelope, and VPR. And then you have your non-structural proteins that are here. So what are these involved in? TAT is required for transcription. REV is RNA transport. WIF is genome fidelity. What that means is how accurate it keeps the genome. And then you have your accessory proteins that tend to enhance HIV production. You have your NEF, VPR, and VPU. Okay? Now, once you make the primary transcript for HIV-1, the splicing generates more than 25 different types of mRNAs, okay? Some of them are singly spliced. They're called the 4 kilobase class. And some of them are doubly spliced, and they're called the 2 kilobase class. So here's there's the full length 9 kilobase transcript, and you have a splicing happening here, and let's say there's a splice site here. So that's your 4 kb class. How much are you removing in this case? You're going from 9 kb to 4 kb. So you're removing about 5 kb of material, okay? Then you also have your another set of splicing here where you have the 2 kb class, okay? So your 4 kb and the full length, they encode for most of the structural proteins, and your 2 kb class mostly encodes for regulatory proteins, okay? But there is some overlap between the two, as you can see right here, okay? So after looking at the genome, how does the HIV enter the cell and replicate? As we learned earlier, HIV-1 targets the cells of immune system by recognizing CD4 receptor, as well as chemokine receptors on those cell surfaces. Your primary receptor is a CD4, and it's found on both T lymphocytes as well as monocytes or macrophages, okay? But it also needs a co-receptor. It could be either CCR5 or CXCR4. R5 is basically for viruses that use CCR5, and this is macrophage trophic. So what that means is if you have CCR5, chances are it's a macrophage, and that's what you will infect. And X4 is T cell trophic. So this is the viruses that bind to CXCR4 receptor, which are found on T cells. The variations in the viral surface protein, or GP120, it determines what kind of co-receptor you are using and what kind of cell you will infect based on that surface protein variation, okay? If you were to flood the system with ligands for these receptors, they can actually block viral entry. Why is that? Because here's your CD4 primary receptor, CCR5 secondary receptor or co-receptor, and here's your surface proteins. If I were to add ligands for these viral receptors or these host receptors, what's going to happen?
The receptors would be like internalized, and they wouldn't be able to interact, or at least occupied, and wouldn't be able to interact with the virus.
Okay, so if you add all this excess ligand, what's going to end up happening is those receptor sites will be occupied, okay? And as a result, the virus will not be able to bind those receptors, okay? Those receptors might be internalized after activation. That normally happens in the cells, okay? So it can actually block viral entry. Now, if we continue along these lines, what we see is that your CD4 binding actually causes major conformational changes in your viral GP120. It's a glycoprotein, okay? And this conformation change exposes the regions that recognize the co-receptors, and it also exposes the GP41, which is the fusion domain for these proteins, okay? And this fusion domain then inserts itself into the host membrane, just like we have seen for other viruses. So here's your CD4 binding, conformation change, and exposure of GP41. Once the viral and the host cell membranes are in close proximity, the fusion will happen, and this will release the viral core into the cytoplasm, as you can see here. Once inside the cytoplasm, the core becomes loose, it partially breaks down, and it permits the virus an access to the cellular material, so cellular nucleotide pool, for example. Because remember, to carry out reverse transcription inside the core, you need nucleotides. Viruses don't make nucleotides, so they have to rely on the host cells to acquire those nucleotides. So your nucleocapsid partially breaks down or becomes loose, and as a result, now the virus has access to the nucleotide pool. The reverse transcriptase replicates the viral genomes, but it generates small mutations, as we saw, because it does not have the proofreading activity. And finally, you can see that it can affect targeting by the immune system itself or even drugs. Let's say you design a drug for one variant, but because of these mutations, the region you designed the drug for has changed. As a result, the drug won't work anymore. Okay? Similarly, your immune system cells may not recognize if the change is drastic enough, or over time it becomes drastic enough. Okay? So as a result, current therapies actually use multiple drugs that target the various steps in virus replication. Okay, a lot of these, they tend to target reverse transcriptase as well. Okay? But unlike other retroviruses, HIV does not wait for the cell to divide. It actively directs the transport of proviral DNA into the cell nucleus. So as a result, you get a productive infection even in non-dividing cells. So you have your matrix, VPR, and integration, or integrase, sorry, which is a part of your pre-integration complex, and that directs the transport of the whole integration complex into the nucleus. Because your matrix, or MA, has a nuclear import signal. It interacts with proteins that bring cargo into the nucleus through the nuclear pore complex. These proteins are called importins. Okay? Your VPR and integrase, they interact directly with the nuclear pore. So here's your HIV core with the genome, and it's going to basically insert itself or gets transported inside the nucleus through this nuclear pore complex. And from there on, the DNA will integrate itself into the host genome. So what needs to happen before you get to this point, you need to carry out your reverse transcription and then send the DNA in. Okay? So if you were to try and eliminate HIV, so the cure for HIV, it's fairly complicated because of this integration. Your immune system cannot identify infected cells if there's no viral proteins on their surface. So if the viral proteins are not expressed, your immune system is not going to recognize that there's an infection. And as a result, you sit latent, the virus sits latent for a long period of time. So after the proviral DNA is integrated, you either get a latent infection or you get an active infection. Okay? This is regulated by transcriptional control elements in the long terminal repeats of HIV-1. Okay? Because they can bind various cellular transcription factors, so NF-kappa B, NF-AT, okay? And these sequences, they exist in the U3 region of your LTR. And these transcription factors, they can be activated by multiple different types of cellular stimuli. They are transcription factors, so they control, for example, NF-kappa B controls a whole bunch of genes in our cells. So for example, let's say if you engage the T cell receptors on the surface, it's a stimulus for T cell activation. The T cells become activated. This results in activation as well as nuclear translocation of NF-kappa B. And since it's a transcription factor, it regulates a bunch of genes inside our genome, okay? But at the same time, there are sequences in the LTR that get recognized by NF-kappa B, and it carries out the transcription. So due to this latent infection, the patients must continue the treatment for their life, okay? So that's the difficult part, because as soon as you stop taking those medications, your viral load will go back up, and it can affect your immune cells, obviously, and you're more likely to pass on the infection if you're not careful, okay? So again, let's look at the transcription of HIV. So you have a viral TAT protein that gets localized to the nucleus. TAT stands for trans-activator of transcription, okay? It's a highly basic, 86 amino acid protein. Why basic? Yeah.
The side, oh, like, it wasn't like the side chains have a lot of like NH2.
Okay, so it has a lot of amino groups, the side chains. But what's the key thing about basic proteins under cellular conditions? Are basic side chains. Remember, they are positively charged under those conditions, and DNA is negatively charged. So they bind very readily. So you get this highly basic protein that's produced by your doubly spliced HIV-1 mRNA. And this protein binds to another sequence, which is called the TAR sequence, which is TAT responsive element, okay? This is near the transcription start site, and it increases HIV-1 transcription by promoting the elongation by your RNA polymerase II. So again, here are your different proteins and your single splice and double splice RNA. But if there's no TAT present, and here's your TAR RNA, your RNA polymerase will start synthesis, but it will come off. It does not stay on the template, okay? But when the TAT protein is present, it will bind to those TAR sequences, and as a result, it will help RNA polymerase stay on the template, okay? And now you can make the transcripts as you wish. So again, your TAT binds to the TAR on the growing nascent RNA molecule, but it does not bind on the proviral DNA. So this is one of the first evidence that there are sequence elements on newly synthesized RNA that actually can detect or direct the activity of RNA polymerase, okay? So this is one of the first evidence that tells us that the sequences that are present on the RNA can actually direct the activity of RNA polymerase. Because when there's no TAT, RNA polymerase lacks what's called processivity, which is the ability for it to travel the entire length of genome. But in presence of TAT, you also get your cellular CDK9 and cyclin, which are required, which are recruited to the complex, and they are involved in increasing phosphorylation of the carboxylic terminal of the RNA polymerase, okay? So CDK9 is a kinase, cyclin is a cell cycle protein, and they're called cyclins because their numbers go up and down depending on what stage of cell cycle you're in. But basically, they phosphorylate the C-terminal domain of RNA polymerase, okay? This happens in our cells as well, where you have to have this phosphorylation for the RNA polymerase to actually work efficiently. So you can actually go read the whole template and synthesize those molecules. Okay? Everyone clear on that? Yes? Okay. Now there's also what are called CRS sequences, or cis-acting repressive sequences. These are present in GAG, Pol, and envelope regions that inhibit RNA transport to the cytoplasm until the right elements come in place. Okay? So you need to have a Rev response element, or RRE, for transport. Okay? So your viral Rev protein mediates the cytoplasmic transport of viral mRNAs. Rev stands for regulator of expression of virion proteins. Okay? So these viral RNAs need, or these Rev proteins need to bind to your Rev response element. So here's the Rev response element, and you have your Rev protein bound to exportin. This is a standard mechanism. Exportin 1 and RAN GTP, they help export stuff outside the nucleus. Okay? So your Rev protein will mediate this cytoplasmic transport that encode the HIV-1 structural proteins. They bind to RRE, and this RRE is only present on unspliced or single-spliced mRNAs. Double-spliced mRNA does not have the CRS sequence, and it can be transported normally outside the nucleus. Okay? So you have this whole complex now sitting on the RNA. This gets transported out, and from there, the protein will come off. The Rev protein comes off. It will bind to importin, come back in, bind another RNA molecule. And now your mRNA is inside the cytosol, where you can start translating into HIV proteins. So this Rev protein continuously shuttles between cytoplasm and the nucleus. It has both nuclear import signal as well as nuclear export signals. Okay? Now, both your TAD and Rev, they are essential for transcription and transport of these mRNAs. So after your proviral DNA integration, only the doubly spliced 2 kilobase class RNA is transported out of the nucleus directly. And this is what allows for the synthesis of your early proteins, which in this case are TAT, Rev, and NEF. Okay? TAT and Rev get imported into the nucleus, carry out the transcription of the provirus DNA, and export of the RNA that has that RRE sequence. And from there on, you have your full-length or single-splice mRNA encoding other viral proteins. So these will be your late proteins. So what I would suggest is for next class, try to draw a model for this system and see how that would work. Okay? Everyone clear? Okay. Now, if we look at the different proteins of HIV and their function, your VIF protein stands for viral infectivity factor. And this protein increases, as the name suggests, the infectivity of the virion. Fairly small protein, about 193 amino acid, and this is found in the cytoplasm of the infected cells. Now, if you were to delete this gene from the genome, your HIV infectivity in vitro is reduced. So that tells us that this protein has a role in infectivity. And this is also, the various proteins are also required for counteracting your cell antiviral factors. So your cell does produce antiviral factors. So this virus needs to counter that. And this is a specific deaminase that changes Cs to to use. Okay? This protein gets incorporated into virions, and it could deaminate residues in the viral DNA. So it will change the C's to U's. This mutates the viral DNA, affects the viral proteins, and eventually reduces infectivity. Okay? Now, problem with this is when the cell becomes infected, your WIF will induce ubiquitination and degradation of this deaminase. Okay? Because this protein now gets polyubiquitinated, it's going to be degraded by the proteasome, and as a result, you're going to have increased infectivity. So again, here's, if you look at the figure, here's your viral APO3, here's your WIF. Normally what WIF will do is it will result in ubiquitination of your APO3. And that gets destroyed by the proteasome. But this also gets packaged and sent to the other cells. And where you have your hypermutated viral DNA, either that can be degraded or it can be integrated. But the point here is that you need this WIF protein to actually inhibit the APO3 deaminase. Okay? Then you have your VPR protein, which is virion protein R. And it also enhances HIV-1 replication. Okay? Again, fairly small protein, 100 amino acids, and it also gets recruited into the virions where it interacts with the C-terminal of the GAT proteins. And it plays a critical role in entry of pre-integration complex into the nucleus. Okay? Now, one thing that happens here is that this protein actually facilitates the packaging of cellular glycosylase, uracil DNA glycosylase, or UNG. This actually removes uracil from the DNA. It's a cellular repair enzyme where if the DNA somehow has a U added on, this will remove that U and fix it. So your VPR actually packages this UNG that removes the uracil from the DNA. So if the host cell did send its machinery to fight against the virus and convert it from C to U, your VPR converts that U back to C. Okay? So it actually removes the uracil from that DNA. Now your VPR can also arrest infected cells in a G2 stage by promoting the degradation of your cellular proteins that are required to go from G2 to M phase of the cell cycle. And this is beneficial for HIV because transcription is most active at the G2 stage. Okay, this is when the cell is getting ready to divide. It hasn't divided yet, but it has a lot of high activity, lots of transcription. So this is advantageous because this VPR protein can arrest the cell in that stage or keeps the cell in that stage longer. Okay? Everyone good? Okay. Next is your VPU protein, which is viral protein unique to HIV. Scientists are not very creative when it comes to names, but there's a very systematic naming convention. So you have your VPU, and this basically enhances the release of progeny virions from the various infected cells. Again, small protein, about 81 amino acids, and this gets inserted into the membrane through its N-terminal domain. And this protein tends to accumulate in Golgi as well as endosomes. Your CD4 in the cytoplasm can also bind and retain the viral GP160 protein. This is the precursor to the viral GP120 protein. And as a result, it reduces GP120 incorporation into the virions. But the VPU that's present here in the membrane can actually trigger degradation of CD4 through its binding to a specific protein, which is beta TRCP. This releases the GP160, and it increases the surface expression of your GP120 and GP41. This is the cleavage products of your GP160. Okay? This VPU also counteracts host antiviral factors that tether the virus to the host cell surface. So some of these antiviral features will basically grab onto the virus and won't let go. So it can't go and infect other cells. So this VPU, it actually counteracts those steps. Okay? So again, here's your GP160, here's your CD4 receptor, and if it's degraded, that's fine. Otherwise, it will counter these your antiviral factors. Okay? So you're going to process this GP160 to make GP41 and GP120. Okay? All right. So how does this work? Remember, VPU counteracts the host antiviral factors that tether the virus to the host cell surface. So this actually enhances the virus release from the plasma membrane. Okay, normally the cell will hold on to this, but in this case, this will counteract those effects and increase the release or enhances the release. Because if you don't have VPU, virions will just tend to accumulate on cell surface. So if you were to take out the VPU gene, the virions just accumulate on the cell surface. And if you were to express HIV VPU1 or HIV1 VPU, it also enhances the release of other unrelated viruses. So how does it works is it basically induces degradation of a protein called tetherin. Okay? And as the name suggests, it tethers these virus particles onto the host surface. So here's your virus particle, it's budding, here's your tetherin, and here's your VPU1 protein, or VPU protein from HIV. This will actually degrade these connections, okay, and the virus particles will then be released. But as you can see, there are other viruses here as well. So in this case, there's a Ebola virus, glycoprotein, SARS-CoV. They all tend to inhibit tetherin dimers. Okay? Now, this VPU is also involved in NF-kappa B activation, so in the inflammatory response. And tetherin actually promotes endocytosis and degradation of virions. So if you were to degrade tetherin, that means you are interfering with all these different processes. Okay? So you're not only enhancing the release, but you're also interfering with the host antiviral strategies. So the virus is basically trying to bypass the host trying to kill it. Okay? All right. Next is the NEF protein, which is the negative effector protein. And this is an important mediator of your pathogenesis as well as enhancing the infectivity of these viruses. Again, small protein, about 210 amino acids, that's localized to the inner surface of the plasma membrane through a myristoyl modification. Okay? So you have a 14-carbon saturated fatty acid, which is myristic acid, which gets attached to the N-terminal amino acid of this protein. And this protein then works to decrease the surface expression of CD4 receptors. And that works through another protein called adapter protein, or AP2. Also, it interferes with MHC1 localization. And both of these proteins are important mediators of the immune response. So again, here's your NEF, here's your MHC, and AP1, AP2 in this case. So if it were to go into an endosome, it will be degraded. So you're modulating the signaling and you're modulating this CD4 receptor degradation. Okay? But the end result is that you're modifying T cell signaling in these cells, and even without the presence of an antigen, you're activating these cells. And as a result, they cannot mount an effective immune response. Okay? So here's your CD4 receptor. NEF is binding to that. It's going to recruit AP2. And as it does that, it's going to help internalize the CD4 receptors through the clathrin-coated pits. And as it does that, your CD4 will go to the endosome and degrade it. Okay? Everyone clear? Yes? Okay, let's see. One more slide and I'll let you guys go. Okay? Lastly, we look at the psi signal, which is the genome packaging signal. This is encoded only on the genomic RNA. Okay, it does not get, because it gets removed once splicing takes place. So there's intramolecular base pairing, which forms secondary structure, and it causes the psi sequence to be inactive. Okay, but as you form the genomic RNA dimer, remember RNA is stored in 2 copies in these virions. So it's a dimeric molecule. So when the dimer forms, you expose this psi sequence. And this now can interact with the nucleocapsid protein, and that can trigger assembly of the virions. In addition to all that, you have your several GAG proteins that interact with the nucleocapsid genomic RNA complex, gets transported to the plasma membrane through the S-cut complexes. Okay? And lastly, once the virion is released, the proteases in the virus particle are activated. They cleave the GAG and the GAG-Pol polyproteins into individual peptides. They self-assemble into the virion to form this conical capsid. And as a result, the virus is now active for an infection. So here's an immature HIV particle, and here's a mature HIV particle. So again, as an exercise, look at the differences. What are the differences between immature and mature HIV particles? Okay? All right. Here's the life cycle. So if you can explain all the steps, you understand how HIV works and how retroviruses work. I know I've given you a lot of information today, but we needed to finish this. And next week, we'll start with double-stranded DNA viruses. Okay? So don't forget, your quiz 7 is going to be available Wednesday at 10 p.m., and you'll have until Friday, 10 p.m. to finish it. It covers both lecture 7 and 8. Okay? Because remember, lecture 7 was fairly short. So it covers lecture 7 and 8. All right. See you guys later. Just wait for me outside because there's a class.